Hi everyone! We are Group 34 here to tell you about the project we've been working on for the semester.
Our project follows a disease called Duchenne muscular dystrophy, or DMD, which we will tell you more about later in the video.
Something you might not know about Duchenne muscular dystrophy is that females can be carriers and can be affected mildly, but it typically only affects boys.
This is due to it being an X-linked recessive trait.
Now, to tell you more about our disease,
Joel Chamberlain, in the article Progress Towards Gene Therapy for Duchenne Muscular Dystrophy, describes Duchenne muscular dystrophy as a genetic disorder of progressive muscular degeneration and premature death.
The DMD protein is 969 amino acids long and 2,907 nucleotides long with a molecular weight of 111.2 kilodaltons.
The gene codes for the protein dystrophin, which is responsible for connecting the muscles and skeleton and keeping the muscle from wasting away. The DMD gene, one of the longest human genes known, measuring at 2.3 megabases, is on the short arm of the X chromosome at position XP21, and according to Primerblast is located 31,097,677 bases and 33,339,441 bases on the reverse strand. Due to being located on the X chromosome and the fact that DMD is a recessive trait, in order for someone to have Duchenne muscular dystrophy, all the X chromosomes must contain the affected allele.
Our gene actually does undergo alternative splicing. Out of the 30 transcripts of DMD, there are 20 transcripts that are protein coding. Furthermore, out of those 20, there are 2 alternatively spliced transcripts, DMD 201 and DMD 227, which are responsible for actin binding. Both DMD 201 and DMD 227 have thousands of variants, some of which have no relevance to our disease at all, such as breast tumors or cardiovascular abnormalities. Due to the fact that DMD is one of the longest genes found in humans, it makes sense as to why there are many different transcripts produced that code for different proteins and therefore have different functions.
Dystrophin is a cytoplasmic protein which connects muscle fiber across the cell membrane to the extracellular matrix, contributing to the structural integrity of the muscle. In addition to skeletal and cardiac muscular support, dystrophin also plays a role in the chemical signaling within cells, according to the U.S. National Library of Medicine.
When there is a mutation in the DMD gene, the gene fails to fulfill its duty in the transcription and translation of the protein dystrophin, which is what causes Duchenne muscular dystrophy. Essentially, Duchenne muscular dystrophy is caused in the majority of cases by deletion in the dystrophin gene, DMD. The disease is an X-linked neuromuscular disease typically caused by disrupting the reading frame in the dystrophin gene, according to the study of deletion mutations in Duchenne muscular dystrophy in Western Saudi children.
When cloning our gene, we first found the nucleotide sequence of DMD. Then we used the nucleotide sequence to find our longest open reading frame. After, we used reverse transcriptase to obtain the cDNA, or the complementary DNA necessary for the PCR template. Subsequently, we used PCR to amplify our open reading frame to ensure our gene was the one being amplified. To do so, we first found a forward and reverse primer outside of our open reading frame. You can see the original primers we chose via Primerblast. After, we then re-amplified our gene with the new primers we designed using a restriction enzyme we chose to be NDE1. This is an integral step of cloning due to the fact that with our newly designed primers, we can insert our gene into the PMOL-C5X vector between XMN1 and NDE1. Our new primers are shown here as well. When expressing our protein, we used E. coli to express our DMD gene via inserting our plasmid inside the bacteria, also known as transforming bacteria. We tested to ensure that our protein was being expressed by using the PMOL-C5 plasmid and labeling our protein with maltose binding protein, or as we refer to as the tag. When purifying our protein, the labeled protein, or the protein with the tag, was purified from lice cells using affinity chromatography. As a result, the purified protein can be seen on an SDS page gel, where proteins are separated by size, similar to a gel electrophoresis. Here, our protein with the tag, or maltose binding protein, was shown to have 1,356 amino acids with a molecular weight of 153.7 kilodaltons.
The first motive for cloning genes may be to gain information about the nucleotide sequence of the gene. DNA sequencing, or restriction enzyme cutting analysis, can be used to study a gene or compare versions of a gene from different sources. A second motive would be to manipulate a gene. This means to alter the gene's DNA sequence or combine different DNA molecules together.
And that concludes our project and presentation about DMD. We hope you learned something new.
No.